your 5 o'clock news, and we're coming to you straight from Fort Wayne, Indiana.
We have some breaking news for you tonight that you won't want to miss.
So stay tuned, because this is information that just might save your life.
Nipah has found its way into the United States, and here's what you need to know. The virus itself varies in size and is encased in the lipid membrane. It is a single helical strand of RNA with 3 proteins wrapped around it, which are N, L, and P. The NLP provide polymerase for replicating the negative strand RNA. There are 2 different proteins on the outside of the viral envelope. The G protein attaches the virus to the host cell, and the F protein attaches the host cell and injects its contents into the cell to create more virions.
Nipah virus was first identified in 1999 in Malaysia, where it had first spread from fruit bats to pigs. In Malaysia, there are thousands of pigs because pigs were their main source of income. And once the pigs were infected, the pigs started spreading them to people. Now, the slaughter of nearly 1 million pigs temporarily solved the issue, but a mutated strand of the Nipah virus reemerged in Bangladesh a few years later.
The Nipah virus is distinct in the fact that the mutated copy that reemerged in Bangladesh had the ability to be transferred from person to person. In addition to this, it's also distinct because it causes encephalitis in humans. And I'm here with Dr. Brick to discuss the Nipah virus. Dr. Brick, what are the symptoms of the Nipah virus and how does it spread?
Well, there are 2 strains of Nipah virus. You have your Nipah M, which originated in Malaysia, and then you have your Nipah B, which originates in Bangladesh and India. The Nipah M virus was spread from bats to pigs and then horses, and then to humans, while the Nipah B virus is just direct human-to-human contact. The exact route of this transmission is unknown due to the new onset of this disease, but it is believed that the Nipah virus is spread through aerosol droplets because the epithelium within your respiratory tract is the initial target site for viral replication. Early stages of the infection with Nipah is characterized with a flu-like illness that can progress into pneumonia and acute respiratory distress syndrome. Changes in the respiratory tract include hemorrhage, necrosis, and inflammation of your small airways. In the late stages of infection, the blood vessels are targeted by the virus and result in vasculitis, blood clots, and vascular microinfarction of the central nervous system, lungs, spleen, and kidneys. The infection then spreads to the central nervous parenchymal cells where it induces an encephalitis response, which is characteristic of the disease. The exact mechanisms for all of these ailments as well are unknown due to the fairly new discovery, like I said before. We know the incubation period of Nipah is between 5 and 14 days, and the symptoms will start to present themselves between 3 and 14, with fever, headache, followed by drowsiness, mental confusion, and disorientation. Early stages of Nipah show pulmonary conditions that will eventually lead into your neurological symptoms. Symptoms can often progress into a coma between 24 and 48 hours, And all of these symptoms are characteristic of encephalitis that Nipah produces. It is possible to have a long-term sequelae from Nipah, but with consequences of persistent convulsions and personality changes. And latent infections have often been reported to reemerge months and years after initial exposure and cause death.
Wow. And Dr. Brick, how would you diagnose this disease?
Well, a diagnosis can be made within the early stages of the infection with a number of tests such as real-time polymerase chain reaction from throat and nasal cultures, cerebrospinal fluid, blood and stool samples, as well as urine. But if a diagnosis needs to be made in the later stages of infection, an ELISA antibody test needs to be done to check for the antibodies.
Okay. And how do our immune systems play a role in trying to ward off this disease?
Well, our innate immune response is activated when Nipah virus genomic RNA is recognized by our cellular cytoplasmic RNA helicases to activate interferon responses. The Nipah produces proteins P, V, W, and C, which they all inhibit interferon production within the cell, thus allowing time for viral replication. Little is known about our adaptive immune response due to the lack of sufficient animal models, and it is known, however, that just 4 days after exposure, the immune system has measurable levels of IgGs and IgMs, which are the antibodies our bodies produce to ward off infection. And these indicate that both B cell and CD4 T cells are active.
Thank you, Dr. Brick. In order to prevent future outbreaks of the Nipah virus, much more research is needed. It's important for people in countries that were previously exposed to the Nipah virus to be aware of the signs and the symptoms and take immediate action in order to prevent the spreading of the Nipah virus. The vaccinations, immunizations, and treatments of the Nipah virus are extremely limited at this time. This is due to the fact that this is a complex virus that has emerged fairly recently, and also the research is very limited because it is a biosafety level 4. This means that it requires the highest safety measures, and it is only able to be studied in a limited number of labs throughout the whole world. In addition to this, a mechanism of the activation of the virus remains very uncertain at this time. This limits the researchers' ability to develop a drug that targets this specific virus. As far as vaccination goes, research is being conducted on the use of a subunit vaccine that may help fight against the Nipah virus. This subunit vaccine uses the G protein without presenting the viral particles of the Nipah virus, and it provides antibodies against the Nipah virus. This has been tested on horses in Australia, but researchers believe that it has potential to be very useful for humans as well. As far as the immunization goes, human monoclonal antibodies may be efficient in immunizing against the Nipah virus. These antibodies also target the G glycoprotein. This method has not been approved for the human use yet, but it has been tested on a variety of non-human primates, and it has resulted in the survival of many of these primates that it has been tested with. For treatment of this disease, researchers have high hopes for the drug ribavirin being used. It's an antiviral drug that is currently used to treat hepatitis C. The mechanism is not totally understood behind it, but it's believed that it controls the mRNA capping. So far, this ribavirin use has not been approved to be used in humans, but in the future it may prove to be effective. But however, as of right now, the only way to treat the virus is with the common supportive care. And while providing this care, extreme caution is to be taken in order to prevent the spreading of this disease. In the future, researchers should definitely focus on the G protein that is involved in the Nipah virus. That's because this protein contributes to the attachment of the virus, and it has already been the basis of a lot of potential vaccinations and immunizations regarding this specific virus. If a drug were developed that blocked the initiation of this G protein, the spread of the Nipah virus in the body may be able to be stopped.
As pharmacist Allie mentioned before, Nipah virus isn't easy to study because of its biosafety level 4 accommodations, but a recent study was published in 2010 where squirrel monkeys were infected with the Nipah virus and observed for 2 months. And then during that time period of 2 months, blood and tissue samples were collected to check for the Nipah virus within the tissue and blood samples. RNA extraction was performed to look at the Nipah virus in the squirrels' genomes. The main techniques of PCR analysis was chosen to track down the Nipah RNA. The blood samples were also taken in order to check for the antibody presence within their blood. The squirrels showed signs of infection 7 to 19 days after being exposed to the virus. And symptoms included acute respiratory syndrome and uncoordinated motor function, which very highly correlates to those symptoms that are found in humans after they were infected. So some of these squirrels were exposed intranasally because the Nipah was thought to be spread via aerosol droplets in previous epidemics. But these squirrels that were exposed intranasally did not have RNA in their tissues, but only the antibodies in their blood. Although the intravenously affected squirrels were detected for Nipah RNA in all the tested tissues. And this shows the quick replication cycle of the Nipah virus. So the progression of this virus matched that to the patients that were affected nearly 20 years ago. So the squirrels proved to be a good model of the human, and further studies could be done to find an effective treatment method with the use of these squirrels. And because of the quick spread of this virus, any suspected outbreak should be reported to the CDC immediately. And almost almost immediately as it is noticed, it's called an epidemic because of its quick spread between humans and animals.
As a cautionary measure, farm workers should be extra cautious around their animals because it is an animal host virus.
If an infection is suspected, it needs to be reported to the CDC immediately.
And since we are going into cold and flu season, let's just keep up our ordinary hygiene protocol. Wash your hands. Thanks for tuning in and have a good night.